Today we will learn about the diagnosis and control techniques of plant prokaryotic diseases. Prokaryotes, a group of unicellular microorganisms with a prokaryotic structure and a nucleus without a nuclear membrane and no fixed form. The species include bacteria, actinomycetes, and cell wall-less mycoplasmas. Some of these bacteria and phytoplasmas can cause a variety of important diseases and are called phytopathogenic nuclei. General traits of phytopathogenic prokaryotes. 1. The morphology of bacteria are spherical, rod-shaped, filamentous, spiral, etc. Most of the phytopathogenic bacteria are rod-shaped, and most of them have long and thin flagella. Gram-stain reaction of plant pathogenic bacteria is mostly negative, and a few are positive. Bacteria reproduce by fissiparism. When a cell grows, it divides transversely from the middle to form two daughter cells. Phytopathogenic bacteria can be cultured artificially, and colonies are mostly white, off-white, or yellow. Phytoplasmas have no cell wall, no gram-stain reaction, and no other accessory structures, such as flagella, mainly divided into phytoplasma and spiroplasma genera. Transmitted by grafting and insects, infected plants often cause yellowing, dwarfing, bunching, and organ deformities. The main groups of plant pathogenic prokaryotes from a practical point of view. Eubacteria can be divided into three phenotypic groups, namely gram-negative bacteria, gram-positive bacteria, and mycoplasmas. There are 6 main genera of gram-staining negative bacteria. 1. Pseudomonas spp. Can cause plant leaf spot disease and leaf blight. A few species cause wilt, rot, and tumor and other symptoms. 2. Xanthomonas genus. Most of this genus are plant pathogenic bacteria, causing plant leaf spot disease, leaf blight symptoms. A few species cause wilting, decay. 3. Soil bacillus genus. This genus of bacteria can cause plant tissue expansion, tumor formation. 4. Owenbachia. This genus of bacteria can cause decay or wilt of plant tissues. 5. Raoul's bacteria. This genus of Raoul's bacteria can cause wilting of Solanaceae. 6. Sclerotinia spp. This genus of bacteria is limited to parasitic in the sclerotia and cannot be cultivated artificially, which can cause citrus yellow dragon disease. 2. Gram-stain positive bacteria. Important genus in the genus Corynebacterium. Mainly cause wilt symptoms. 3. Mycoplasma. The bacterium has no cell wall and is surrounded by protoplasmic membranes, which can cause mulberry dwarf disease, puffball bush disease, etc. Phytoplasma disease symptoms and characteristics. I. Bacterial diseases. Plants infected with bacteria can produce necrotic spots, rot, wilt, tumors, and other symptoms. II. Symptoms include discoloration and deformation, including yellowing, dwarfing, or dwarfing branched leaves, small leaves, flowers, etc. In addition to mycoplasma diseases, most bacterial diseases can be diagnosed by the phenomenon of spraying bacteria. The diagnosis of phytoplasma diseases is to observe the bacteria with the help of an electron microscope and to look at the characteristics of the disease. Transmission of phytoplasma diseases. Bacteria usually invade the host plant through wounds and natural orifices, such as water holes and stomata. In the field, bacteria are spread mainly by running water, including rain and irrigation water. Phytoplasmas are often spread by insect vectors, such as leafhoppers, grafting, and codling moths. Control methods for phytoplasma diseases. The first control measure is to strengthen quarantine, rice, leaf blight, bacterial streak disease, citrus yellow dragon disease, etc. are phytosanitary objects. So such diseases to strengthen the quarantine of the transfer of agricultural products to be quarantined by the relevant departments. Second, the establishment of disease-free nursery. Cultivate disease-free seedlings to ensure disease-free seedlings. To ensure that new orchards are disease-free, new seedlings do not carry bacteria. 3 is to strengthen pest control. Timely control of spreading insects and field pests. Fourth, the choice of disease-resistant varieties. The choice of high-yielding disease-resistant varieties suitable for local cultivation can reduce the occurrence of disease. 5 is to strengthen cultivation management. First, strictly prevent string irrigation, diffuse irrigation. Timely drainage of the field to avoid partial application of nitrogen fertilizer. What's more, the orchard should be cleaned to reduce the base of the disease. Sixth, chemical control. Seed disinfection can be carried out before or at the beginning of disease onset in the field. Choose suitable pesticides to control different diseases, such as Mesophilus, zinc thiazole, allicin, copper preparations, etc. Thank you for watching. We will see you in the next